Welcome to our second topic. It's quite logical really because we've looked at cells and now we're going to stick them all together and make some tissues. There's 4 different types of tissue. The first one we're going to talk about is epithelial tissue. Epithelial tissue is quite major really when you consider it covers and lines and it forms the glands, so it can be very varied. We're not going to go through all of them, but we will look at the principal characteristics and the classifications of epithelial tissue, and we will look at specifically the epidermis, vascular endothelium, enterocytes and hepatocytes. And we will look at those specifically because it either feeds into clinical reasoning or they are very important epithelial tissues. Our second tissue that we'll look at will be connective tissue, and we're only going to look at this briefly because connective tissue is relatively inert, so we don't really need to come back to it in any detail at any point. So in terms of connective tissue though, we have connective tissue that you're probably all thinking about and can see pictures of in your head. The sort of fascia and the cartilage, that sort of thing, the packing tissue. But connective tissue has specialized connective tissue. And these include some pretty different ones that you wouldn't think were connective tissue, like blood. So we'll look at that in a little more detail. Our third one we'll look at is nervous tissue. Now, we do have neurophysiology further on, so we're not going to look at it in too much detail, but we will start to talk about neurons as the main cell of nervous tissue, but there are other cells involved as well, and these are the support cells within the peripheral nervous system and different ones in the central nervous system. And we will start to introduce the concept of afferent and efferent pathways in preparation really for neurophysiology when we talk about it in more detail. Finally, we will, in this session, we'll start to talk about the types of muscle tissue. There are 3 types, so we'll look at a comparison really of the 3 different types and a little bit of detail about cardiac muscle and about smooth muscle. Cardiac muscle, of course, we'll come back to in a more functional sense when we look at cardiovascular. And smooth muscle is quite important in terms of not only the organs, but also lining all the blood vessels. And finally, we'll introduce skeletal muscle in terms of comparison with those other types of muscle. And then in part 2, we start to get a bit more dynamic because we're actually going to excite some skeletal muscle and then contract it. So part 2 will be a little bit more dynamic. This is really, in this part, about talking about those different types of tissues and categorizing them and comparing them. So as we said, there are 4 primary types of tissue: connective tissue, epithelial tissue, muscle tissue, nervous tissue. It's not about the cells because each tissue may contain different types of cells, or the predominant cells within that tissue may have different characteristics depending on their location. And that's what we're really going to start to compare in this section. So let's start with epithelial tissue. We can call it epithelial tissue if we're talking about a single epithelial tissue. Then we can call it epithelium. That's the singular for the tissue. If we're talking about lots of epithelial tissues, then we can talk about epithelia. So, as I said right at the beginning, we have 2 types of epithelia really. that we can differentiate in terms of function. The first one is covering and lining. This forms the boundaries between different environments. And hopefully you're already thinking in your head about possible environments and possible types of covering and lining epithelia. So we've got things like the skin, of course, and the lining of hollow organs. So the mucosa and extending all the way inside in terms of every single hollow organ. So the GI tract, the respiratory tract, the vascular system, which is obviously a closed system, but is also lined with epithelial tissue. Urinary tract and the reproductive system. So these are the coverings and linings. The other type is glandular, and this is what forms all of the glands of the body, including all the endocrine glands that we're going to talk about later in the module. Overall, the functions of epithelia, it varies according to location, but overall, if we stick them all under the umbrella of epithelial tissue, we've got protection. So, for example, the skin and the mucosa. We've got absorption. So, enterocytes lining the digestive tract. A key function of those, of course, is to absorb nutrients from the lumen of the GI tract. Filtration and excretion. So, for example, in the kidneys and the renal tubules. And if we're talking about glandular epithelia, secretion, because the whole point of glands is that they secrete things like hormones. All epithelia, though, have the same characteristics. So, our characteristics, the big one is apical basal polarity. So in other words, there are distinct differences in the structure and the function between the top and the bottom, the apical and the basal surfaces. And if you think of covering and lining, that would be fairly evident, wouldn't it? I was talking about enterocytes. They absorb. They're going to absorb from the top, but then they need to do something else with it at the bottom. So we have an extreme difference between the top and the bottom. At the apex, there may be transport mechanisms and receptors, or there may be cilia. At the base, there is always the basement membrane. And we'll talk a little bit more about the basement membrane in a sec. Another characteristic is specialist junctions. And there are a few of these different junctions. We're going to talk about the main ones. So we have to hold these epithelial cells together. And again, if we're talking about covering and lining, of course, those junctions have to provide some sort of fairly strong seal, but also a little bit of give. So first up, closest to the top, closest to the apex of epithelial tissue, we have tight junctions. And these prevent the passage of proteins, and they also maintain apical basal polarity. Adherens junctions are another type of junction you may come across. These initiate and maintain cell-to-cell contact. And that cell-to-cell contact can be very important, particularly in terms of proliferation of epithelial tissue. Desmosomes are another specialist junction. Desmosomes resist shearing forces and mechanical stress. So they have little elastic fibers between the cells that can move, and a bit like those bridges that have the little zigzaggy bits either end. so that in a high wind, that bridge has got a little bit of give, or when it gets cold, it can shrink. When it's hot, it can expand, etc. These desmosomes, though, can be unzipped. And this is another key thing in terms of moving cells around and also proliferating more cells within a space. Hemidesmosomes are half desmosomes, and they actually secure the epithelial cells to the basement membrane, make sure it doesn't drift off. Gap junctions are more like channels, really. They're not so much junctions as like a little channel between two adjacent cells. So they can open and close, and this allows things like ions and other solutes, glucose, to pass directly from cell to cell. So they don't have to go out of the cell or enter each cell individually. They can flow between cells. So we talked about tight junctions before, and we talked about adherens and desmosomes and gap junctions, and here they all are. And in epithelial tissue, they are very important. The basement membrane is another characteristic. So the basement membrane of epithelial tissue is really what gives it the basal part of the apical basal polarity. So the basement membrane is fixed to epithelial cells, as we said, by hemidesmosomes. And this often provides an additional barrier. We have sort of two parts to it, really. We have the basal lamina, which is closest to the epithelial cells, and that's divided into the lamina lucida and densa. That's not terribly important, and they're named really because of how they appear in a microscope. The basal lamina is secreted by the epithelial cells, which is logical because that's the layer that's closest to them. Below that, we have the lamina reticularis, or the reticular lamina. And this is collagen fibers secreted by the underlying connective tissue. So that forms the basement membrane. All epithelial tissues have a basement membrane, but probably the one that you're most aware of is the border between the epidermis, which is epithelial tissue, and the dermis, which is connective tissue. Other characteristics: epithelial tissue is avascular. In other words, it doesn't have its own blood supply that extends into the epithelial tissue. There are no capillaries running through epithelial tissue. So it's avascular, but it is innervated. So in other words, it has nerves. We know this because of paper cuts. You get a paper cut and you'll look at it and there's not much to see and it doesn't bleed. That's because you've only cut into the epidermis, the epithelial tissue. There is no blood supply there. If you cut a bit deeper, of course, yes, it will bleed. But paper cuts really hurt. That's because the epidermis, being epithelial tissue, is innervated. It has sensory neurons. It has nociceptors that detect pain. So it's supplied by nerves, innervated, but it doesn't have blood vessels. It is avascular. In terms of solutes, because of course epithelial cells still need nutrients, oxygen, etc., etc., those solutes diffuse from blood vessels within the underlying connective tissue. as we said, hence paper cuts hurt because of the innervation, not innovation, but they don't bleed because epithelial tissue is avascular. Another key characteristic is regeneration. Epithelial tissue regenerates very well. If the cells are damaged, they will proliferate very easily. And this is logical, really, because if you think about it, covering a lining, it's going to be very prone to damage from friction, attack by pathogens, inflammatory processes, acids, irritants, all sorts of things. So if you do damage it, it needs to recover quickly. So it needs to proliferate new cells to fill the gap. So epithelial cells have a high regenerative capability. But this normally is limited by contact inhibition. In other words, you've got an epithelial cell. Either side of it, it's got an epithelial cell joined to it by those specialist junctions. So if you like, it's holding hands with the adjacent cell. If you take away that adjacent cell, it's no longer holding hands with anything. And that will stimulate the cell that's no longer holding hands to proliferate, to go through the cell cycle, to make a new cell to fill the gap. So it's limited by contact inhibition because we've got cells butted up against it. But the minute you take one of those cells out, it will start to regenerate. So when junctions or adjacent cells are removed, the cells proliferate via mitosis very easily. The problem is this also gives a high risk of malignancy, as in carcinoma if it's epithelial tissue, because they have a tendency to go into uncontrolled proliferation. If the checks and balances that we'll talk about in clinical reasoning during the cell cycle are ineffective, then you get uncontrolled proliferation, which in its own right is not a good thing, but stays relatively benign potentially until such time as it breaks through the basement membrane. And if you think about it, the blood supply is not within the epithelial tissue, it's in the connective tissue, so it's the other side of the basement membrane. As soon as those cells can get into a blood supply, then of course they can shoot off to other places and metastasize. The other thing with this uncontrolled proliferation is some people are prone to overgranulation. And we've all seen it, haven't we? If you've done any wound care of any kind, you come across someone and the epidermis is not closing off. It just keeps proliferating. And this is because their contact inhibition doesn't work very well and they've got uncontrolled proliferation. So it's not necessarily malignant, but it is uncontrolled proliferation. What we usually do, of course, these days, gone are the days of silver nitrate, I hope, but what we do these days is we try and apply pressure, don't we? You try and apply a pad and lots of nice, strong elastoplast to push on it. And what you're doing is you're mimicking contact inhibition to try and stop that proliferation. So we've talked about characteristics. Here we're going to talk about classification. The first way to classify it is by cell shape. So squamous epithelial cells are flat, like pavement slabs, and because they're flat, they've got flattened nuclei. Cuboidal is, as it suggests, they're cube-shaped. and they have round nuclei. Columna, again, as the name suggests, are tall. They're like columns. They have oval nuclei and they often have cilia at the apex at the top. The other way to classify is by number of layers. So we can put that together with the cell shape classification. So simple epithelia is a single cell layer on a basement membrane. Stratified epithelia, here we've got layers of cells stacked one on top of each other, and the basement membrane then sits at the base of the stack of cells. And you can see at the top there we have stratified squamous epithelium. So we've got squamous shape cells with multiple layers. It's not simple, which would be one layer. It's stratified. It's got multiple layers, and right at the bottom, it's got a basement membrane. Below that, we've got stratified cuboidal, stratified columnar, and then a rather funky one, which is pseudo-stratified columnar. And if you look carefully, it is actually only one layer. So it is technically simple, but it looks stratified because it's got a bit squashed up and all the nuclei are different heights. The shape and the number of layers often relate to function. So, for example, the epidermis is stratified squamous epithelium because we've got multiple layers for protection. The respiratory membrane across which the gases diffuse in the alveoli is very, very fine because things have to diffuse across. Endothelium as well. Both of these are simple squamous epithelium. In the case of blood and lymph vessels, although it is epithelium, it's referred to as endothelium. Endothelium is a specialist term for the epithelial cells that line blood vessels and line lymphatic vessels. So these very thin layers allow for the movement of solutes and in some cases cells between different areas. So some specific examples then. So the epidermis we've talked about as being stratified epithelium. New epithelial cells are pushed up from the base, from the stratum basale, so the basal layer, and right up to the top. And as they go up, they become cornified or keratinized. They change and lose a lot of their functions, but they also become able to hold water and plump out the skin and provide a protective layer. So at the bottom we have the basement membrane, of course, and this forms the border between the epidermis and the dermis. Above that we have these new baby cells, if you like, and then those gradually move up through the stratum spinosum, the stratum granulosum. Here they're now starting to develop little granules within them that retain water. Stratum lucidum you actually only get in very hard skin areas like the soles of your feet. And then finally at the top we have the stratum corneum. And it's the stratum corneum that the cells then slough off from. So the basement membrane forms the border with the dermis. Vascular endothelium, So as we said, this differentiates the epithelium that lines blood vessels and lymphatic vessels. The term endothelium is also simple squamous. These simple squamous epithelial cells and the basement membrane form a layer in blood vessels called the tunica intima. And we'll come back to that in clinical reasoning when we talk about where atherosclerotic plaques form. Endothelial cells in the tunica intima, of course, are in direct contact with the blood. So they need to be fairly permeable because we need to get things out of the blood and into the tissues. And you'll notice below that we have smooth muscle cells. We'll talk about smooth muscle a bit later, that enable vessels to vasodilate or vasoconstrict by contracting or relaxing. In terms, though, of the endothelial cells, we have quite a lot of permeability. This is because the way these pavement slab type squamous endothelial cells lie down, there are gaps where they don't quite meet together, they don't quite tessellate. And of course, that can be changed if those endothelial cells contract. If they contract, those gaps get bigger. If those cells relax and become even more pavement slab-like, then they close the gaps a little bit. So permeability can be regulated by changing the shape subtly of those squamous cells. In some vessels, depending on the levels of permeability required, the endothelial cells themselves have pores through them called fenestrations. This would be fenestrated endothelium. So there are actually holes through the cell to increase the permeability. In some cases, they have an incomplete basement membrane, so the basement membrane has big gaps in it. And this happens in sinusoidal endothelium, like the sinusoids within the liver that we're going to talk about in a minute. And in some cases, they allow something called transcytosis. So in cell biology, we talked about transport and we talked about endocytosis and exocytosis. In transcytosis, what we've got is endocytosis on one side, so this would be on the blood side, and exocytosis, so it travels through the cell within a vesicle and is exocytosed at the base. So something completely moves through the cell by endocytosis and exocytosis. Enterocytes. These are simple columnar epithelial cells that line the ileum and the colon. There are slight differences between the ileum and the colon. The enterocytes and basement membrane form villi in the ileum. So we've got these finger-like projections covered in enterocytes, epithelial cells, projecting up and down into the lumen of the ileum. At the bottom of those finger-like projections, the villi, you have crypts. And both those areas really have different functions we'll talk about in a minute. The colon, though, doesn't tend to have villi. We're really talking about the ileum if we're talking about villi. So at the top of the villi, the enterocytes are more absorptive. Their role is more about absorbing whatever is in the ileum, in the lumen of the ileum that, of course, we've taken in, we've eaten, digested. And then they can absorb that through various transport mechanisms. The enterocytes in the crypt, which are actually the younger ones, are more secretory. Because that's logical, isn't it? They're tucked away miles down away from the lumen. They're not going to be very good there at being absorptive. But when we get to clinical reasoning, we'll talk about one of their secretory roles in terms of fluid regulation within the GI tract. So our enterocytes that line the crypts and line the villi are joined, held together by selectively permeable tight junctions. I know you'd think tight junctions, they're not going to be permeable. But in various places throughout the body, tight junctions have to be selectively permeable to allow some things through. And this is one of those examples. The ones particularly in the crypts are semi-permeable, the tight junctions, to sodium and water. And this is what I was saying about fluid regulation in the lumen. This is all part of that. So the tight junctions do allow some things through. The enterocytes also have short cilia at the apex. So it's a good example of apical basal polarity because there will be a basement membrane underneath the enterocytes as they go up to the villi and in the crypts, but at the apex they have cilia, and this cilia form the brush border. And we've got the same situation really as we had with the epidermis. The enterocytes in the crypts are the immature ones, like the ones at the basal layer of the epithelium. These are immature in the crypts, but they're also secretory. And then they gradually move up the villi, they become more mature, but also more absorptive. Eventually, just like the epidermis, they get to the top and they become senescent and they slough off into the lumen. And of course, more immature enterocytes are made in the crypts and move up the villi. When they do get to the top, so the enterocytes, the villus enterocytes, they need different transport mechanisms to absorb lots of different things. You think of all the things you eat that need to be absorbed. And those things, depending on their size, will have different transport mechanisms. You can see in the diagram there. For example, we've got large peptides and proteins are being endocytosed, whereas sodium is being moved in via a transporter. So they have different transport mechanisms in order to absorb water, electrolytes, in other words ions, carbohydrates as monosaccharides once they've been broken down into monosaccharides, proteins once they've been broken down into small peptides and amino acids, and fat. Fat is absorbed as monoglyceride or free fatty acids. Once they've been absorbed, they're transported through into the basal side, and up each villi we have a blood supply. So they go through the villi into the centre, into the veins that go up the villi, which eventually drain into the hepatic portal veins. Or in the case of fat, that goes into the lymphatic system. The hepatic portal veins and the lymphatics all then go off to the liver. The liver is the next organ along the pathway, if you like, along the circulatory pathway. So they go off to the liver, and the liver's job is then to process or store whatever nutrients we've taken in and transport it off to the liver. which brings us nicely, really, to hepatocytes. So remember, we did say that tissue, if you're talking about a tissue, we're not definitely talking about one cell type. And the liver is a good example of this. 80% of the liver cells are hepatocytes. So they are epithelial cells. Because they're epithelial cells, the other characteristic, of course, is that they're highly regenerative. The liver is able to regenerate quite well. The hepatocytes, though, are really highly complex, and we don't have time to go through all of the functions, but there are many metabolic and endocrine and secretory functions. And this includes, of course, the metabolism and the regulation of all those carbohydrates, lipids, and proteins that we sent there from the enterocytes. Hepatocytes also have microvilli that help to increase absorption. And they're organized into lobules. These lobules are hexagonal. And the lobules have a central vein. They have a branch of portal vein on the outside of the lobule, and also an arterial supply, because after all, they need nutrients to keep themselves going. But this blood supply, the arterial and the portal blood, then drains to the central vein down these little channels that are like blood vessels, basically. They're lined with endothelium still, called sinusoids. That drains into the central vein, and then ultimately will go off back to the heart via the inferior vena cava. So they receive blood from the GI tract via the hepatic portal vein. That enters the sinusoids, the blood vessels within the liver lobules that run between the rows, if you like, of hepatocytes. And a bit of arterial blood also, because, of course, hepatocytes need their own supply of oxygen and everything else to keep them going. The blood flows through the sinusoids. Meanwhile, the hepatocytes, of course, are in contact with it, so they can absorb from that blood. The blood then enters the central vein, as I said, goes back to the heart, and into the heart via the inferior vena cava. Which brings us to connective tissue. So, okay, we said at the beginning, we have proper connective tissue, and this is the sort of connective tissue that you might imagine. It's cartilage in your earlobe, it's all those membranes that bind other tissues together, it's the dermis, all that packing stuff, adipose, areolar tissue, all that is connective tissue. It's packing, it's relatively inert though. And then we have specialized connective tissues. So connective tissue, particularly proper connective tissue, forms an extracellular matrix. And this extracellular matrix is then produced by cells that are found within that matrix. So in terms of different types of connective tissue, we have different cells that produce it. Fibroblasts we'll come back to in immunology. They're responsible for healing, basically. Fibroblasts secrete connective tissue proper. So they secrete collagen and elastic fibers. And there's a lovely picture there. The blue blobs are the fibroblasts, but the green fibers are what they're secreting. Chondroblasts secrete cartilage. Osteoblasts secrete bone. The term blast really refers to immature cells or cells that produce tissue components, whereas site is a common word for cell. You will come across it repeatedly, this whatever site. This really is referring to mature cells. So as we said, connective tissue can be proper connective tissue or specialized connective tissue. Our specialized connective tissues are things like bone, cartilage, and blood. In terms of our proper connective tissue, we can then start dividing that down depending on its function and its location. So we can have loose proper connective tissue, and this is really the sort of padding type stuff. Areola, reticular, adipose, reticular meaning a network that supports things. We can also have dense proper connective tissue. And this is more about tendons and ligaments, so it's dense. It might be elastic, and it has a more sort of dynamic function, if you like. In terms of specialized, as we said, we have bone, blood, and cartilage. Again, cartilage can be divided into different types as well. We have elastic, hyaline, and fibrocartilage, depending on whereabouts it is in the body. Bone, of course, we have spongy and compact, and generally spongy tends to be immature, disorganized bone that hasn't been organized into nice little round osteons that all fit together. Blood we'll talk about in a minute. So our loose proper connective tissue is about cushioning and supporting, so whether it's supporting cells, supporting the skin, supporting vessels, or even whole organs. It's also about nutritional reserve, particularly if we're talking about adipose tissue. So you have lipid vacuoles in adipocytes. That's why you get basically fatter, because you develop more adipose tissue if you have an excess of these fats that you haven't burnt off, of course. And also warmth and protection. As we said, the dense proper connective tissue, this is the stuff that's much more organized, more dynamic, forms the tendons, the ligaments, fascia, that tough membrane around organs, organ capsules, the dermis, plus also the elastic fibers of the arterial and the bronchial walls. Blood, yes, is a very atypical connective tissue. So let's talk about blood briefly. It's slightly different in terms of the connective tissue because instead of having a more gel-like matrix, we have a liquid matrix, a fluid matrix, and of course that's plasma. Plasma is mainly water, but within it there are also quite a lot of plasma proteins. Not plasma membrane proteins, bear in mind, plasma proteins. So these might be things like globulins that carry other things. We also have ions, electrolytes, hormones, endocrine hormones in particular have to get all the way around the body, so they have to use the circulation to travel. Oxygen, of course, glucose delivered to the cells. So there is sort of 10% of the plasma is made up of these solutes. The rest is all water. Of course, it doesn't contain fibres like other connective tissues. Otherwise, we'd have an awful lot of occlusion and clots. But the formed elements, so the equivalent, if you like, of things like collagen within the matrix, the formed elements, are the cells, the blood cells, and platelets. So, of course, we have erythrocytes, red blood cells. We have about 48 to 54 billion per milliliter of blood. So, because there's so many of them within blood, they make up about a quarter of all body cells. White blood cells, of course, the amount varies. Hence, we do blood tests, don't we? We look at white cell count. More on that in immunology. But generally, on average, we have about 7.5 million per mil, so a lot less. Leukocytes, again, we'll come back to this in immunology, but it includes neutrophils, monocytes, lymphocytes, also the granulocytes, known as mast cells, eosinophils, basophils, that sort of thing. They're all white cells, leukocytes. Platelets, although they're called thrombocytes, they're not really technically cells. They're cell fragments, really, because they're anucleate. They don't have a nucleus. And they are really the cell fragments that then aggregate to start off the clotting process. So if we have a question and I ask you, for example, what a mast cell, what type of tissue does a mast cell belong to? It's connective tissue because it's blood. And there we can see lots of erythrocytes, little disc-shaped red cells. We will find out at some point why they're red. Monocytes, quite big, get even bigger when they get into the tissues, and they become macrophages. Neutrophils, eosinophils, they're granulocytes, so hence they've got that funky inside, and basophils. And the platelets, you can see little tiny cell fragments. That brings us on to our third type of tissue, nervous tissue. And again, as we said, this isn't all about a specific cell type. In nervous tissue, we have a lot of neurons, but we do have some support cells that have vital functions as well. So we can differentiate these in terms of the central nervous system, so the spinal cord and the brain, and the peripheral nervous system. The peripheral nervous system is basically everything else. So that includes the point where the spinal nerves come out of the spinal cord outwards. So we can differentiate it in that respect. Neurons, of course, generate and conduct action potentials. Neurons are the cells within nerves, or most of the cells within nerves. They are individual cells. They generate and they conduct action potentials. They are irritable cells. Remember when we talked about membrane potentials, we talked about irritable cells and their resting membrane potential. So these are irritable because they conduct action potentials. Afferent neurons conduct action potentials towards the central nervous system. So this is our sensory nervous system, really. This is feeding in to the central nervous system, whether that be the spinal cord and or the brain. Efferent neurons are the ones that conduct action potentials away from the central nervous system. So generally we're talking about motor neurons. They're sending signals out from the brain or from the spinal cord to the rest of the body. So afferent in, efferent out. We can also differentiate neurons as to whether they are myelinated or unmyelinated. And in this lovely wiggly worm picture here, you can see that the one on the left is unmyelinated. The one on the right is myelinated, those beigey lumps, sausage-like shapes along its length are areas of myelin that insulate the axon of the neuron, and they make the conduction speeds much faster. It's called saltatory conduction. More on that a bit later. But you can see in the diagram, if you compare the action potentials going down them, it's a lot slower conduction in unmyelinated rather, neurons. Within the central nervous system, neurons form two types of tissue differentiated by colour. We have grey matter. I'm sure you've all heard that. Why don't you use your grey matter? Grey matter is the cell bodies, the somata. Soma singular is the cell body of a neuron. Somata is plural, cell bodies of the neurons, and also their synapses with other neurons. So the grey matter is really where it all happens. The white matter is about axons. It's about the long stretch that transmits, that propagates the action potential along the length. So we have white matter and grey matter. And the reason it's white is because most of those axons are myelinated. And myelin is a bit white. And these follow in terms of the central nervous system, the axons, along tracks. Tracks really are the central nervous system version of nerves. In the peripheral nervous system, we have bundles of neurons together forming nerves. In the central nervous system, those axons bundle together within tracks. So within the peripheral nervous system, all afferent and efferent neurons are bundled into nerves. So in a nerve, we will have action potentials being conducted in opposite directions and to different places. If you think we only have so many spinal nerves, they then branch out and branch out and branch out and branch out and branch out. That's all those neurons packed together coming out from the spinal cord are then getting divided up and sent off into different nerves. In terms of the cell itself, the neuron, they can vary in structure, and this usually depends on really their function. So we talked above about somata, the cell bodies, and the position of the cell body will vary according to the type of neuron. So in the one on the left, we have a bipolar neuron. The cell body is in the middle of the axon. The one next to it, the cell body is in the middle of the axon, but it's on a little stalk, and this is a classic sensory neuron. Interneurons we'll talk about when we get into neurophysiology. Motor neurons are classically multipolar. This means they have a cell body at one end. Sticking out of the cell body, they have dendrites, and at the other end, they have their axon terminals. It's usually the type of neuron that we draw when we're talking about propagation and generation, because they're a lot more straightforward. Even more complex, of course, are pyramidal neurons. Pyramidal cells or pyramidal neurons are within the central nervous system, and they're highly complex with lots of dendrites, lots of fingers sticking out of the soma, the cell body and one spindly little axon. That's because within the central nervous system, they need to make lots of synapses, lots of connections with lots of other neurons so that we can have those complex pathways that enable the higher centers to function. Well, that was neurons. On to support cells. Support cells are just that. They're non-conductive, but they have vital functions that help to support the neurons. So they either support, insulate, or protect the neurons. Schwann cells, we talked there about myelin and how myelin makes conduction so much faster. Schwann cells are what secrete myelin around myelinated axons within the peripheral nervous system. So the cell will bind to the axon, very immature age, will start to secrete, a bit like a roll of cling film really, myelin around itself and that section of axon. And it will gradually increase. So the myelin, the thickness of the myelin sheath does increase as you mature. Glial cells is a bit of an umbrella term for the cells within the central nervous system. It's often used to talk about astrocytes specifically, but technically it covers all of the central nervous system support cells. So one example is our microglia. Every tissue has its own macrophages that sit within it, ready for any dead or dying cells, any pathogens, ready to do a bit of a clean-up job, a bit like little tiny hoovers within the various tissues. Within the central nervous system, the macrophages are known as microglia. So they're found in the brain and the spinal cord. Oligodendrocytes, fantastic word that, they're actually the central nervous system equivalent of Schwann cells. They secrete myelin around central nervous system axons, but they tend to do it on multiple axons. So what they're trying to do is hold all the axons together. So they'll reach out and they'll wrap myelin around one axon, then an adjacent axon, and they help to hold them all together and myelinate the axons. Astrocytes are, quite amazing really. And there's been a lot of research about astrocytes, and this is possibly why the term glial cell tends to be used specifically for astrocytes, because they are so important in terms of the functioning of the central nervous system. So they have multiple functions. They're generally found at central nervous system synapses. They're very much involved in the production, the recycling, the regulation of neurotransmitters. And they also help to create the blood-brain barrier. So they have lots of functions that can be quite complex. Another type of glial cell, ependymal cells, line the ventricles and the spinal cord central canal. And these are the cells that help to produce cerebrospinal fluid, CSF. Talking about our blood-brain barrier. So if we look at a normal blood vessel, remember when we talked about endothelium, we talked about big gaps because we've got this thin layer, we've got big gaps between the endothelial cells. So most blood vessels in the body allow the passage of solutes and even cells between the endothelial cells. In the blood-brain barrier, we have more protection than that. We control and very closely regulate what moves out of, for example, the meningeal vessels into the brain. And this passage is regulated by tight junctions. So we have tight junctions between the endothelial cells that are quite highly regulated as to what they will let through. Around that and around the endothelial cells, we then have a type of cell called a pericyte. Pericytes are only really found around the blood-brain barrier. They can expand and contract, and this depends on signaling mechanisms as to whether they are currently expanding or contracting, but they can regulate the permeability of the blood-brain barrier. So it's not quite such a barrier as you might think. It can change the permeability across it. And then, of course, we've got endothelial cells, so we have a basement membrane. The basement membrane surrounds the endothelial cells on the inside, on the central nervous system side, and the pericytes. In addition, we talked about astrocytes before. We have the astrocytes around the blood-brain barrier form these feet, these projections that wrap themselves around the vessels travelling through the brain and also help to regulate permeability, help to form a barrier, but a barrier that can be selectively permeable. Which brings us to our final tissue. So we're going to talk about muscle tissue, and there are three types. We have skeletal. This is the muscle tissue that allows us to move, that is attached via tendons to our bones and allows us to move, but also in other functions as well. If you think about the muscles of the thorax that enable us to breathe, they're all skeletal muscles. We have smooth muscle. Smooth muscle we came across before around the blood vessels underneath that tunica intima of endothelial cells. We have a tube, if you like, of smooth muscle. And that is also replicated in terms of organs and in terms of the GI tract. You have these tubes of smooth muscle. And cardiac muscle. Well, the only place that's found, of course, is the heart. So let's do a bit of a comparison. So here we've got skeletal, smooth, and cardiac. Okay, the first way we can differentiate them is whether they're voluntary or involuntary. Voluntary, of course, means that we are controlling it. Our higher centers in our cortex are regulating it, are stimulating it when we decide that we want to pick up a pen or walk. So skeletal muscles tend to be voluntary. They're controlled by efferent neurons coming out, remember efferent, that carry action potentials from the motor center. Involuntary, in terms of smooth muscle, this is regulated by the autonomic nervous system and also some other chemicals that can bind and can regulate it, usually fairly locally. So examples like histamine that will come across in immunology, angiotensin II will come across in cardiovascular, and nitric oxide in both, basically. So chemicals can regulate it, but also the autonomic nervous system. So our autonomic nervous system is what controls, what regulates the functions of our body without us having to think about it. Hence, it's involuntary. Cardiac muscle is also involuntary. Here it's actually regulated by the autonomic nervous system. So the autonomic nervous system does have an impact because it can increase heart rate, it can increase the strength of contraction or reduce it. So there is an autonomic input, but the actual main stimulus is from autorhythmic cells within generally the sinoatrial node. The sinoatrial node leads. It has autorhythmic cells that we'll talk about in cardiovascular that will spontaneously depolarize and cause cardiac muscle to contract and relax. Of course, not something we want to have to think about. We'd be a little bit busy if we had to keep thinking about our heart beating. So it's involuntary. So skeletal in terms of overall function allows movement of the body, but also the respiratory muscles. Now you're probably thinking, hang on a minute, she just said that skeletal muscle is voluntary, but we don't have to think about breathing, so we don't have to think about respiratory muscles. So actually, yes, in the case of respiratory muscles, they actually have autorhythmic cells, a bit like the heart does, that will spontaneously depolarize, that keeps us breathing when we're not thinking about it. So they also have some autorhythmic controllers as well. Of course, we can stop it for so long. We have some control of it, voluntary control, but yeah, it's also automatic. But generally, in terms of body movement, of course, in terms of skeletal muscle, we make a decision as to whether we're going to move our arm, move our leg, twitch an eyebrow, anything. And that all takes skeletal muscle. Smooth muscle, as we said, is found in the walls of hollow organs, blood vessels, the bronchi, the GI tract, all those sorts of places. Cardiac forms, of course, the myocardium. In terms of appearance, skeletal muscle is striated. It means it's got stripes across it. So you have these parallel myocytes, muscle cells. There's that term cyte, myo, muscle, cells. You have these parallel long myocytes. If you think of large muscle, the muscles are quite big. The myocytes run end to end. So they're quite long. But they are striated. And all these parallel striations line up and give us a striped appearance. And those striations are caused by little units called sarcomeres, which we'll talk about in the next video. Smooth muscle is non-striated, and the myocytes, in this case, form sheets. They form sheets, and between them they have gap junctions. Remember our specialist junctions? Here we've got gap junctions, those little channels between the myocytes that allow the flow of ions from myocyte to myocyte to myocyte to myocyte very, very quickly. And this means they can contract all at one time. Cardiac muscle also has gap junctions between the cardiomyocytes, but in this case, we have some striation. They're faintly striated. This is because they have the same internal structure, these sarcomeres, as skeletal muscle. The difference is we don't have long parallel myocytes all packed together, so the striations don't quite line up. So the myocytes, cardiomyocytes, branch, so they're not quite so regular. So those striations, those sarcomeres, don't line up, so we get faint striation. But as I said, they also have gap junctions because, again, we need a free flow of ions from myocyte to myocyte to myocyte because we want our myocardium to contract in a regulated way and all together. Otherwise, obviously, we'd have some fibrillation. So yes, we'll talk about skeletal again in part 2 because we're going to talk about excitation, contraction coupling. So a little bit about smooth muscle. As I said, we've got these arranged in sheets. So they're long, tapered myocytes. So they're arranged in sheets with little gap junctions between them. Smooth muscle found in the walls of hollow organs, except the heart, of course. That's our cardiac muscle. Also found in the bronchi and around blood vessels, forming the tunica media of blood vessels. They don't have striations because they have a totally different internal arrangement in terms of myofilaments. The contractile filaments within myocytes are called myofilaments. They're usually primarily composed of actin and myosin. In the case of smooth muscle, they're not arranged in these nice little sarcomeres. Instead, they're arranged sort of on the outside of the smooth myocytes and fixed to them by things called dense bodies. So in a non-contracted and a relaxed state, we have this thing that looks a bit like a net over our myocyte, but when they contract, they cause the myocyte to pull in and also slightly twist. So they have a different mechanism of contraction, not one that we're going to revisit. As we said, this thing about the gap junctions is that it means that ions, specifically cations, so positive ions, generally sodium and calcium, can flow through from myocyte to myocyte very, very quickly. Because they do that, they all tend to contract as one. So this contraction is called syncytial. Syncytial contraction means that contraction is as one thing, all at the same time. And that's because of the gap junctions. So sodium and calcium can pass quickly from myocyte to myocyte to myocyte through these little channels. Everything contracts simultaneously or syncytially. And this type of contraction tends to be, certainly in terms of smooth muscle, slow and sustained. Contraction in smooth muscle, as we said, can be stimulated by the autonomic nervous system. More on that again in cardiovascular, but also local chemical stimulation. Things like histamine. Histamine we'll talk about in immunology, because if we have an inflammatory response, one of the first things that happens is vasoconstriction. So we need those smooth myocytes around the vessels to contract. Nitric oxide also regulates on a local basis. That's what paracrine means. It's very local basis. The contraction and relaxation of our smooth muscle. And hormones. So if you think of the uterus, oxytocin causes uterine contractions. So we've got an autonomic nervous system, but we've also got chemicals that can do the same job. Cardiac muscle, as I said, we've got branching cardiomyocytes, which then form the myocardium. Between the branches, we've got things called intercalated discs. At the intercalated discs, we have the gap junctions because they're then joining one branching cardiomyocyte to another one. Because they've got gap junctions, they also contract simultaneously or syncytially. But in this case, the arrangement is different to smooth muscle in terms of contraction because they contract in exactly the same way as skeletal myocytes because they have the same internal structures of sarcomeres. As we said, the faint striations, of course, are caused by the sarcomeres that are very, very clear in skeletal muscle because they're all nice and parallel and packed together. Here, they're not lined up because of this branching arrangement. So the striation appears faint. So that was part 1 of our tissues and muscle topic. Now you've watched part 1, you can download the PDF or print it, whatever works for you, that goes with it, that reflects the content. Access any suggested resources, but don't forget to join your live session as allocated, where we'll go over it with my trusty whiteboard. Also, remember to make your own notes, ideally portable notes that you can take with you, so in those spare minutes when you've got nothing better to do, you can get a little bit of studying in. Make your own diagrams, add to your glossary any terminology that you've learned in this session, and start to try teaching the topics to someone else. Ideally, someone who's a colleague on this module, or someone who's done the module before, or someone that's actually holding your notes so that they can correct you if you do get it wrong. And I'll see you in part 2.